Okay, this is my biology assignment for Michael Sands in biology, and my volunteer today is my dad. So, show yourself. Stand right here. Say hello.
Hello. Mark's his dad. I'm the photographer. So if the shot's not good, it's not his fault. It's my fault.
Alrighty. So, what we're going to be learning today is protein synthesis. And first I'm going to introduce some things. So protein synthesis is basically the creation of protein, and it all occurs in our eukaryotic cells. And this is an example of a eukaryotic cell. And this purple circle right here, this is the nucleus. And the nucleus consists of nucleic acids, which has two, which is your deoxyribonucleic acid and your ribonucleic acid, which is DNA and RNA. So you'll have single strands and double strands in there. The DNA is a double-stranded, and the RNA would be the single-stranded. So your DNA is composed of 4 nitrogenous bases, and is the adenine, thymine, cytosine, and guanine. RNA is replaced with uracil, which is the replacement of thymine. So anytime you see, like this would be thymine, all the purple ones here. Over here, when this is the RNA. When it's replaced, it gets replaced with the uracil right there. So all the little dots right here, these are all codons. And codons are basically what the DNA gives the cell's instructions. So these will all be codes for your DNA to make a certain type of protein. So the protein could be for your hair, or it could be for maybe your skin or anything. So the really important thing to know about DNA or protein is protein makes up every cell structure in your body, mostly of it. So protein is very important. So each DNA has a leading strand and a lagging strand. So let's say, for instance, the leading strand is here. When it opens up, the RNA is going to come and copy the leading strand that's been opened up. So it attaches, and then it comes out as mRNA. So for each three pairs right here, these are codons. So each has a codon. So when we see in the mRNA, this becomes AUG is a start codon. So anytime there's an AUG, when it goes into production, and we'll see that after as I explain translation, this becomes the start codon. So this is when protein can start making its function and start producing proteins when it hits AUG. And all the N codons, so when it hits an N codon, it would be UAA, or UGA, or UAG. Those are all the N codons. So I'm just going to introduce amino acids. Amino acids need to be, basically for a protein to be created, it needs to be mixed with the start codon, going through the chain, and being mixed with an amino acid. So an mRNA message, a messenger of the code, mixed with the tips here, which are amino acids, that will give you a protein. Okay, you can edit that. Okay, so now I'm going to explain transcription. For transcription to occur, it occurs in the nucleus, and what essentially happens, the double helix of the DNA separates, and a RNA comes in and is separating it, and at the same time it's filtering through the leading strand's message and turns it into an mRNA, which is a messenger ribonucleic acid. and now it comes out. See, this is the same code here as a leading strand for the DNA, and now it comes out as, instead of thymine, it comes out as uracil, so AGU instead of AGT. And also we see here that the starting codon would be here as well, AUG. So while that is occurring, transcription is happening right in here. These are nucleic acids, and this is, it has RNA and DNA in it at the same time. So this would be your DNA being separated by an RNA, turning it to an mRNA, which is the carrier for the leading strand codons. And these little pores I have drawn in here around the nucleus let the mRNA, which is the blue strand here, slip out into the cytoplasm of the cell, where it can there meet in rRNA, which is a ribosomal ribonucleic acid, which helps put together mRNA and tRNA to start protein synthesis. And then we have translation. So now we have translation. When translation occurs, it occurs outside in the cytoplasm, and the mRNA is paired with the rRNA. So this here is basically a larger form of what's happening, or where this strand would be going to. So this is the rRNA, and basically you can see that this strand here has come into the RNA section. It's like a dock almost, and it's going through and it's being read, and basically it's looking for AUG so it can start. Here it's already found it, so the tRNA, which is the transfer ribonucleic acid, and that transfers amino acids. And like I said before, you need amino acids and the mRNA, the message of the codons, to be paired together so it can give it its function of what protein it's going to have, or what protein it's going to be. And so you see the tRNA is going through with the amino acid and it's being dropped off. And at the same time, those three, so there's a 3 here, it will read that code and then it'll be printed onto the DNA, or to the amino acid. And then as it exits out, it doesn't have its amino acid. So this would be a tRNA. This is a larger form of it. It's basically an RNA carrying an amino acid, which is essential for producing proteins. Also, it is essential to know that during translation, the ribosomal RNA, this pink or red part right here, it basically is essential to straighten the mRNA out so it can be read, and then the amino acid can be printed on it. See, as you see, as it exits here, it's all curly and squiggly. So when it's here, it's really straight, and it's going through, and it's straight. Okay, so there's 2 types of mutations. There's point mutations, and there's frame shift mutations. So a point mutation would be if there's a substitution of a single base codon. So here we see the same alignment of DNA up until it gets to the 7th one. So here we see that it's different. This is thymine and guanine. So it would be TG, or just T. So here, the 7th one, 1, 2, 3, 4, 5, 6, 7 is A. And now instead of A, it is T. So this can be a minor mutation, or it can be devastating, because it can make the protein non-functional, and then therefore the protein will have no purpose. Okay. So the second mutation would be frame shift mutation. So I've taken the codons of DNA and I've written it at the bottom here. So as you can see here, it's all the same until it gets to a frame shift mutation. And after the point of frame shift mutation, all the codons will be different. They won't be the same, because it's when there is an addition or a deletion of a single base codon. So here we see the same AGT, ACC, and then we have at the top AGC, and at the bottom ATG. So there's a change. And at the same time, CAA CCA. After the frame shift mutation, all of the codons have been changed after. And that's protein synthesis. And my volunteer, you come around again so I can have proof that you're actually here. Not just... So what can you tell me about protein synthesis? Wait, actually, do you have any questions?
Nope, not really.
No?
Where do you put the nuts and bolts? I'm an industrial millwright, I'm a mechanic.
There you go. And I want to be a nurse, so it's not too bad. But thank you for being my volunteer, and I hope you enjoyed it.
Thanks.